Sure.
And opening the webinar.
Great. Thank you so much. So we can still, keep chatting, and then, but we probably not that many, audience, and then probably many, people also kind of teaching right now also, but typically, some regular folks kind of joining us. I mean, mostly synthetic biology folks, and then mostly people. I mean, right now, watching the YouTube. And then yesterday I got another email from the department chair. His student wanted to, watch this one lively, but the schedule is not good for the student, the class schedule. And then he kind of said, can I share, YouTube? And then of course, I mean, I'm kind of publicly, available all the YouTube, video, except the last week's one, because last week we have the CEO of the company, but he currently on leave from the institute. He's the professor, so he has some conflict of interest type of problem. So we need to release that video on probably next week. But other than that, every video basically out there. And the one interesting thing I gonna observe was I released one YouTube video midnight, and then when I check in the morning, I got already 200 view. That means that is not from US people, that's from Asia, European country, and Australia, and so on. So that means this is really global kind of event right now. So I'm so excited, and then I really thank you for both of you. I mean, you literally kind of make this event success. So really grateful. Thank you so much. Okay, so I'll give a couple of minutes. I mean, do you have any other thing, any exciting thing? Probably beautiful weather, MIT right now, and
today is nice actually. Yeah, it is not that common actually. So yeah.
And also Red Sox win in the Wonder game. I mean, I'm the big fan of Red Sox and also Cardinals here. Unfortunately, Cardinals just didn't go to the off season. I mean the postseason, but right now Red Sox won the amazing game, the two game. Last 2 days or 3 days.
Mm-hmm.
So how about you? I mean, so you are based on the Stephanie. You are right now located in where?
Our office is actually in Research Triangle Park, North Carolina.
Oh, right. So both East Coast.
Yep.
Wonderful. So this time of year kind of best, or when is the best season?
It's one of the nicest times of the year here, yeah.
That's wonderful. Yeah, I mean, same here. Today is not that great. We have rainy day today, but last week was gorgeous. I mean, I even cannot stay in my office. I just wanted to outside and then just have lunch. I have the lunch box always. So bring my lunch box outside and then eat out there. So, okie dokie. I probably want to start because everyone's busy right now. So, okay, let's see. Okay, let's start. So, good morning, afternoon, evening. Hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요. Hello, everyone. Welcome and thank you for joining us, the 8th seminar and US Army Day. By accident, I got to know today is World Standards Day and National Dessert Day. And so probably you want to taste some dessert after your lunch. And also during Korean War, today was unfortunately the Battle of Triangle Hill, which was the biggest and bloodiest battle of 1952. Interestingly, today is also Defender of Ukraine Day, a state holiday, of course, in Ukraine. So why not? So US Army Day from the SynBYSS perspective. I also need to give a talk right after this seminar for US Air Force regarding my US Navy, I mean ONR project. So another coincidence, or fate? Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Stephanie. She works at US Army Research Laboratory as a program manager for biochemistry program. Importantly, she is the program manager for Army Center for Synthetic Biology. I believe many of you submitted your proposal to get funding. And what else do you want to know? Stephanie, take it away.
Thank you so much. First, Professor Moon, I'd like to thank and congratulate you on creating this fantastic seminar series and for your dedication to promoting early career scientists in synthetic biology. And I'd like to thank you very much for the opportunity today to provide remarks on behalf of the Army Research Laboratory. For the Army and the entire Department of Defense, synthetic biology and biotechnology are key research areas, and there are a variety of programs and funding opportunities within the Army and DoD that support research in these fields. For the Army, the key focus is always the soldier now, but also the soldier 20 years from now. When we consider areas where synthetic biology could provide advances for the Army, this is always through the lens of protecting our soldiers, who must conduct operations in the face of extreme physical, mental, and environmental challenges. And so we can think about synthetic biology providing innovative solutions for new and enhanced capabilities with respect to nutrition, physiological monitoring, and medical interventions, novel materials for uniforms, devices, and vehicles, environmental sensors, water purification systems, environmental remediation, and new types of materials for built structures. The opportunities are vast, and the Army needs innovative ideas from the academic and industrial communities to take full advantage of the amazing capabilities that synthetic biology can offer. To this end, the Army Futures Command is spearheading the Army Joint Enterprise for Biological Engineering, which is creating a coordinated and connected Army ecosystem for synthetic biology and biotechnology that is focused on advanced materials and agility in the production and reclamation of materials at the point of need. As Professor Moon mentioned, I am a program manager at the Army Research Office, which is a component of the Army Research Laboratory. The Army Research Office is responsible for executing the majority of the Army's extramural basic research program through high-risk, high-payoff research opportunities that will drive cutting-edge and disruptive scientific discoveries for future Army technologies and capabilities. The Army Research Office is also dedicated to the education and training of future Army scientists and engineers. Specifically within the Army Research Laboratory, there are opportunities to collaborate with Army scientists as part of the ARL Open Campus Initiative. And there are also funding opportunities available. You can find these by searching the broad agency announcements at arl.army.mil. Some of these opportunities include the ARO Core Broad Agency Announcement, which includes basic and fundamental research opportunities across a variety of fields, including the life sciences, biotechnology, and synthetic biology. The ARO Core Broad Agency Announcement includes a variety of different programs, including an early career award program and a short-term innovative research program, which provides support to test a hypothesis or demonstrate a proof of concept. In addition, as part of the Army's recognition of the promise synthetic biology holds for our future soldiers, a new Army Center for Synthetic Biology will kick off in 2022. The broad agency announcement for the center remains open for seedling projects for the next 3 years. This BAA is also available at arl.army.mil, or feel free to contact me directly for more information. Thank you so much for the opportunity to join you today. The research you all are doing today could be the key to the next discovery that protects our soldiers from the threats they face each day protecting our freedom. The future is very bright for synthetic biology and biotechnology, and I look forward to seeing what you all discover. Thank you very much.
Oh, wow, thank you so much for your insightful talk and important information. Of course, I'm exciting collaboration and grand opportunities. So young people just pay attention to her website and then her opportunity. For next few years. Okay, so now the main speaker of today with a longer introduction. Okay, so Eugenia Inda is a pure postdoctoral fellow in the Synthetic Biology Center at MIT. She works with Tim Lu, one of the smartest scientists and engineers in the world, in my opinion. I also worked in the MIT Synthetic Biology Center 10 years ago, and I actually will visit to give a talk next month in that center. She is building biosensors to diagnose and treat inflammatory disorders in the gut, such as inflammatory bowel disease and celiac disease. Specifically, she is equipping the bacteria dwelling in the intestine with sensors to recognize the molecular markers of inflammation and to direct them as sentinels to pester the gut and then secrete therapeutic molecules in situ. She received the 2020 Langer Prize from AICHE for proposing a multi-diagnostic diaper to monitor infants' microbiome development. In fact, there will be in-person ceremony, of course, at AICHE this year, and also reception for her and this year's Langer Fellow Arita. She is also SynBYSS the speaker later on. If you attend AICHE, let's stop by to congratulate, of course, both, Adita and Eugenia. And I will also be there to meet her and also you. Eugenia studied and leads the MIT Microbiome Journal Club with the support of the Center for Microbiome Informatics and Therapeutics at MIT MGH. Besides, Eugenia served as an MIT Com Lab advisor to help students show the value of their ideas and push their scientific career forward. That means she always cared about younger people and the younger scientists and engineer, even though she is also young, scientist and engineer. Outside her research, she loves capturing the beauty of everyday life with her camera. She is in the job market right now and clearly hard to catch or capture. Of course, a science rising star is always difficult to capture. So faculty search committee members in this virtual room, please catch her if you can. Eugenia, take it away.
Thank you so much. Thanks for the kind introduction. And thank you for organizing this. This is really a very exciting opportunity for young researchers. And I know it takes a lot of resources and effort, but we are really grateful for that. So I'm going to share here. Let's see, I want to have this spotlight. From here. Oh. There you go. Okay, so let me move this thing here. Yeah, so thanks for this opportunity. And today I'm going to share about synthetic microbes as next generation diagnostics and therapeutics. I'm here actually now at the Synthetic Biology Center. So then for in the Q&A, I can show you from the window some of the view from here, which is a very nice day today. So actually, I wanted to share first something that wasn't in the bio actually, but it's a fun fact about me. When I first came here to US from Argentina, my name was quite famous actually, but not precisely because of my science, but because of this little piece of art on another plate actually. And to the point that actually a lot of people were reaching out to me thinking that I was an artist and they wanted to collaborate. So that was kind of fun. But I would tell you a little more about the story behind. We were doing a course in Cold Spring Harbor, and there we were modifying genetically yeast in the beta-carotene pathway. So we have all the palette of the colors going from yellow to very orange. We were at the very end given the opportunity to play with different organisms, and then we started doing this painting thing. And then we sent it to the first art contest that ASM was organizing, and my piece of art was one of the winners. The exciting thing of this was that when I first also started sharing, I needed to give a title. So inspired by a very exciting time in Argentina, which is the harvest season, considering that Argentina is an agricultural country, I thought, well, we have all these tools to engineer yeast and all these resources, and it's really an exciting time for research on this. So I give this little part of like, look at the field, like it's ready really for harvest. And I think this is even more true now if we think in the field of diagnostic and therapeutics. So I'm really excited to share about this. I'm thinking that this could be really a time in which people from very different fields will come together with a common goal of using these tools for the health improvement of patients. But I would say that even during the last years has been a challenge actually to revert that, so people to really get to know that I was a scientist instead of an artist. My real goal actually as a synthetic biology is to do something that would be useful, to develop a technology that actually would bring, that would solve real world problems. So I wanted to share this quote that guides my research that says that we need to start by doing what is necessary, then what is possible, and finally we're doing the impossible thing, right? And of course, this is something that we need collaborations and to work together for making this happen. So for me also as a future faculty and as a current mentor, this is something I also seek to empower the next generation of scientists to take their scientific challenges following these steps. First, to be very grounded and sensitive to the needs of the community they serve, to be proficient in their techniques, and then to be imaginative in their scientific pursuits. So I will be sharing a little of what I'm doing now and also what I envision for the future. So I will get directly into one of the areas that I'm most excited about as Dr. Moon was sharing the bio. I'm also part of the Microbiome Center, and there we are sharing, we are part of the journal club in which we share papers and we get excited about the latest news on this. So if you're interested in this, please contact me because we are actually doing this as broad as possible. But so far, I think everyone hear how the changes, the alterations in the microbial composition in the gut have been linked with many different diseases, ranging from Parkinson to asthma to even cancer, depression, and hyperactivity in kids. The reason for this is not so much because of the bacteria itself, but because of the metabolites that they produce. What has been discovered in the last few years is that these metabolites, the problem is that they are produced constantly inside our gut at very high concentration. And they are not kept there. They can actually signal at a distance via the whole circulation, and they can even get to the brain. So they can even cross the blood-brain barrier. So this is how I was really surprised, because how these molecules can really get to basically all the body. And it's not only that they are also so high concentration, reach all the body, but also they can regulate the host metabolic pathways. So they are really very important in regulating our bodies. And so if something is affected, actually, the implications are huge. So this has become then a very exciting avenue for engineering and thinking that, okay, we can change the composition and then we can bring health to patients. But it's not that simple. The question is what really makes a healthy microbiome. And the problem is that actually the gut is like a black box, actually. It's difficult to access. And so we don't fully really understand what's going on there. But even if we could predict the composition, the healthy composition for each individual, that on top of that, for each individual, we understand that would be different. The second challenge is how can we actually reconstitute it, right? How such a complex system can heal itself and reestablish the original healthy composition. So if we think about other ecosystems that actually we understand better, for example, like in a forest, after wildfire, if we want to reestablish the original stratification, even if we know what are the flora and fauna at each level of all the stratification that we can see in a forest, after a wildfire, it's impossible to reconstitute it top down. Actually, the problem is that for the forest to be what we know, each species needed to build codependently on each other for many, many years, right? And so the same thing happens with the microbiome. Each microbial species build on each other. And so it's not something that we can rebuild top down. So what is then the hope? This is why this is a time for prevention, that we really need to work on identifying as soon as possible when the problem is happening. So what we propose is to monitor the chemical environment of the gut so that we are able to detect when the patient is going from the healthy state and getting into a disease state. So inflammation would be like a wildfire, right, for the gut, right? And new research have been showing that antibiotics can cause a lot of dysbiosis and even bacterial infections also. And for example, for particular inflammation that is that a lot of groups are studying, which is called inflammation bowel disease, even though the etiology of the disease has not been determined, it has been, there are evidence that show that there's higher levels of oxidants in the gut. And so we can see higher levels of reactive oxygen species, reactive nitrogen species that start to change the environment. And so then second step, the microbial communities start to change. And this makes a lot of sense then to study this chemical environment, because the chemical environment is the one that determines which metabolic pathways can get activated, both either in the host or in the microbial communities. So once the chemical environment change, then all the metabolic pathway changes, and finally the microbial communities will change, which is finally what actually we see now in the papers, the correlation of different composition of bacteria and how that is linked with disease. But we need to get to the cause to be able to treat diseases, right? The problem is that currently researchers don't have the tools that are non-invasive to do timely diagnosis, to really catch as early as possible when this chemical environment is changing, that leads finally to changes in the microbial community. So we want to focus on this, on timely diagnosis that would be able to understand the causes of the disease and the interaction between the host and the microbial species. Not them independently, but all together. It works actually inside of our guts. So if we think about the current technologies we have, like the gold standard actually for IBD diagnosis or inflammation in general, is the endoscopy. Endoscopy is a highly invasive technique in which the patient actually is in a situation in which, like there's like gases injected so as to open the gut, and so that the gastroenterologist can check the different polyps and different structures inside of the intestine. But this is very highly invasive, it's costly, and it's limited the number of tests that can be performed per year. Alternatively, we have these electronic capsule endoscopes, which are safe. They are much less invasive, and they have been on the market for many decades, and they actually are FDA approved. They are really efficient, but the problem with these technologies is that they only inform images. They only provide images of the places where the pill is going through in the gut, and not the molecules that actually we were describing, that we need to define the chemical environment. And then, of course, yes, we have stool analysis in the lab that they do measure these biomarkers, these chemical molecules. But the problem with this is that these molecules, when they actually exit the body, the most important ones, the ones that determine the chemical environment the microbial communities are exposed to in the gut, they get degraded or absorbed or metabolized by the host. So even if you think that actually the fecal microbiota is only partially representing the real autochthonous microbiota that is in contact with the mucosa. So still a biopsy needs to be done. And even in all these techniques, as soon as you get ambient exposure, for example, to oxygen, the level of oxidation will change. So if we think of all these molecules that we want to measure the level of oxidation, it's actually all these methods are really invasive and are actually destroying that information. So what we propose is, as a next generation diagnostics, is to use bacteria that are commensal bacteria already in the gut and to engineer them as biosensors so as to be able to measure non-invasively the molecular mediators of disease. So I think this is really a smart approach, because if we think that these bacteria are already there, and so they are robust, they know they are really adapted to this environment. We have so far technologies that allow us to do this direct evolution so that we can even optimize them to work better in that environment. And they have natural sensors that we can adapt for our purposes. They can continue sense, and of course we can scale this to even have low cost products. So for example, the very classic thing we do as biotechnologists to all this cloning and engineering, in which here, for example, we have this analog sensor, which is sensing a reactive oxygen species here with peroxide. And so the different concentrations can be measured with a readout here with a fluorescent protein like GFP. The thing that happens with this is that when we put this bacteria inside of the gut, the signal gets messy because the environment is very harsh. You know, like we have very low pHs. The bacteria need to adapt to be able to produce this protein. So in our lab, we have developed some computing functions that allow to improve the signal to noise. So we have this digitization process, which adds a recombinase system, in which once the molecule is detected, triggers a recombinase that flips the DNA and turns on the GFP. So this allows to make the signal like digital, and so that we can have really from 0 to 100% of activation in a very small range of different concentrations, right? So we can play around different promoters, different RBSs, and so we can play with all the different tools we have in synthetic biology to change this. And this allows us to build actually different sensors that can sense at different thresholds of a target molecule. So this allows us to build what I like to call this stage detector, in which we can measure low, medium, and high levels, which would be indicative of a mild, moderate, and severe disease state. So for example, here I'm showing how we have tested this in mice. And so we gavage the mice over multiple days, and we treat them with a model of inflammation, which is treating the mice with DSS. And so we can follow what's going on with different inflammatory markers inside of the gut, just collecting from the stool. So one interesting thing, we can see how in the DSS model, we have this trigger of this high inflammatory signals, but we are able also to identify individual mice that they have a very particular response. The second part of this is that we can really follow what's going on inside of these mice instead of what is currently needed, and we need to do a necropsy and do all the analysis, right? But still here we needed to analyze the fecal samples. So for patients, we thought we could do something different. And so we got inspired by this legendary movie, actually, Fantastic Voyage, in which they have a kind of a similar challenge, in which they needed to remove a blood clot inside the brain of a dying scientist. And so what they do is they reduce the whole surgical task force to the minimum size so they can get in there and remove the blood clot. So we thought, what if actually we shrink the whole diagnostic process to the size of a chickpea? So that we can get inside the gut and from there do the diagnosis. And so here what I'm showing is kind of the big picture of the project, in which when we are able to detect at molecular level what's happening inside the gut. The bacteria are engineered to produce light in response to these molecules once they cross a membrane that allows them to get inside of the bacteria that are encapsulated there. And then we also have electronics that are able to detect that light and produce luminescence. These electronics are wirelessly can transmit information to a smartphone, for example, and then the gastroenterologist can get the information in real time. So as you can see here, we are able to detect this inflammation, and then we will be able to distinguish between the disease state versus a healthy situation. This is also very interesting because this changes from patient to patient, and so the ability for us to also measure different thresholds allow also to see, for example, for one patient, a higher level of a particular biomarker could be a healthy state actually, but for other actually would be higher. So the possibility to measure over time the progression lets us to see the situation of each patient. So one key aspect that we needed to work here was on the miniaturization. And the information we have with other current technologies, for example, the PillCam, have a 1.4 retention that actually in these cases, they need to get into surgery, right? So the minimum size we could get actually will get much better safe possibilities. For example, this Procardia one, we have really very low reported cases in billions of patients. So what we did actually, that was also one of the big challenges coming from already a technology that was developed in our lab by McElhinny, in which he developed this device, but it was at a centimeter scale. And so for here, the power level was milliwatt to nanowatt. So the challenge here for us to minimize this was to get to the nanowatt level. And so as to be able to get also to the millimeter scale. So the other challenge was that for many of these multifaceted diseases, we need to measure multiple biomarkers. It's not only one biomarker that determines the state, that fully diagnose the disease. So we need to add more biomarkers. And so here we are, as a proof of concept, showing 4 biomarkers, but actually we could just probably extend it to 16 or even larger. And so we could play around with different biomarkers, or actually different biomarkers with different thresholds of activation. And so here I'm showing some pictures on how it works. We have here the 4 channels, and then we put the membrane here, and from there the electronics are reading the luminescence. The membrane helps to permeate the molecules, these small molecules we're detecting, and then the electronics transmit wirelessly the information. Here you can see, in comparison with the dime, the millimeter scale that we are working. So here I'm showing what would be really like the big picture of what we are doing. And this is kind of what I propose as a platform for using it for many other diagnostic tools for other diseases as well inside the gut. We have this recombinase system that allows us to test in animal models. And this is important because in mice, for example, we can do engineer mice, and we have like mutants, for example, that are reflecting a particular disease. We can change the diet. We can even do treatments with antibiotics. Many questions that people in the microbiome field are interested in. We can test different constructions of different biosensors that we are developing. And then the ones that are validated in mice or for example, in pigs, and we are also testing this device, can be used in the device. And the device then could be finally used of information transmitted wirelessly to have a follow-up from the gastroenterologist. And actually, we are proposing that if the patient, for example, takes this every day or every week, we can fully have a chemical landscape of this disease. And so to see if the patient, for example, is in a healthy state and after an antibiotic treatment, probably it triggers a redox imbalance. And so now it's falling into a disease state. And then the therapeutic can be provided, or changing lifestyle or diet could be proposed for them. So then one of the questions that was missing was like, well, this helps a lot to see in real time. But talking with some gastroenterologists, it's like we need to know more at the localized level. And for example, for Crohn's disease, it's important because inflammation happens in patches. And so what we want is actually to be able to follow over the days what's going on there. And then also with the promises during therapeutics, so the bacteria will be able to detect and respond, right? And so what we developed here was this magnetic hydrogel living device in which we encapsulated the bacteria inside of a flexible, tiny hydrogel that can be swallowed and gets into the gut, and then is attached to a particular region of interest with a magnet that is attached to the abdomen. So we have been doing this with the mice. So I'm excited to show our results in vivo with this. So I will share some of the challenges that this technology, developing this technology presented. We did this in collaboration with the SAO lab here in the MIT SynBYSS Center, and they are experts in material science. So the challenge here has been a very, very collaborative research because we needed to work with two challenging things that happens in the gut. If we think different from what happens in the skin, that the bacteria are not in such a harsh environment. On top of that, this has the complexity of the movements of the pressure that happens in the gut. So we have this peristalsis that helps to actually move the food bolus along the lumen, but we wanted actually to retain the magnetic hydrogel. So we needed to play around the magnetic force so that to be able to retain the bacteria in a particular region. And also we have in the intestine the segmentation, which helps to disintegrate the food bolus. But this also is a threat to the integrity of our magnetic device. So we also needed to make them really tough to be able to resist this segmentation. So for this, there has been a really very collaborative project in which the SAO lab has been working on the material side. So for example, here we need to test how these ferromagnetic metals that we were incorporating in the hydrogel together with the bacteria, because we see both things together in the hydrogel, and then we solidify everything together, how that could affect actually and could be even toxic for the cells, for the human cells, right? So we know this is the level of toxicity, the minimum level of toxicity. So we tested how inside the gut, so with simulated gastric fluid and with intestinal simulated fluid also, that the concentration of both metals should be below this level of toxicity. And then we check with Keiko cells how that actually impacted on the viability, and we see that after 7 days, everything is okay. And then for me, this was really very exciting as a microbiologist, because it was really challenging also to keep the bacteria alive during all these days. And the reason for this is because when they are encapsulated in these hydrogels, also the level of oxygen that they have access to is limited. And so we needed to engineer the right level of bacteria that would allow to use as much of the nutrients that they needed, but at the same time to be able to give a signal. And so, as you can see here, we were playing around at different concentrations of basically how tough the magnet, the hydrogel needs to be, so as to be able to resist all the pressures inside the gut, but at the same time to keep the bacteria alive. So after 7 days, we could say that we could succeed on getting the bacteria viable, and at the same time with the level of toughness that the hydrogel needed for the gut. And here we're showing how it actually works with the mice. So we attach here the magnet to the abdomen, and we can see here the different hydrogels that they get retained to the lumen of the gut. And so this happens over 7 days. We could see the retention there. And then we wanted to see the functionality. So we use here a blood sensor that was developed for this cool project of the embed, this device of the wireless transmission of information. So we use this now for the magnetic device. And so we use this sensor that can detect blood, and we induce the bleeding inside the gut with indomethacin. And after a day, we could see the signal coming from the magnetic device. So this shows that the bacteria are not only alive, but they also are able to function, to be functional, to detect the bleeding and to give the signal that we need. And then finally, I wanted to share actually what is more of my projects for the future as a future faculty. And I have this opportunity to participate in the Langer Prize for Innovation and Entrepreneurial Excellence. So I wanted actually to share here how the American Institute for Chemical Engineers are bringing this opportunity from this, doing a world of good, which are resources actually for young researchers, which I think this will be very exciting for this audience to participate and to propose these very high risk, high impact challenges that young researchers have been thinking of. So this is like a seed funding that helps to start this line of research. So for me, this has been something that I've been thinking for many years. I like volunteering, helping in activities with kids, especially from church and in some mission trips. And one thing I really like from kids is that they are like mini scientists. They are curious. They are always like asking questions and like trying to understand things in a very with a very, how to say, a naive way of thinking, right? Like really being open to whatever they would encounter. However, in one of my trips in the north of Argentina, really actually in the same more from Bolivia, we were working with some communities there, and I found that in particular one community, some kids like didn't respond to the normal stimulus that kids like responded, like games, they were not engaged, and it was very, very hard to communicate and to see really ways to engage with them. And I was wondering, okay, what's going on here? Like, what is the problem? And in this trip, actually, we were helping also many doctors that were providing basic health care. I was like talking with doctors, what's the problem here? And I felt totally helpless because there was really no way to have any diagnosis of the water or the health state of the kids and why actually they were not responding to the basic stimulus that we were bringing. So what I thought, it would be great if we could actually develop diagnostic tools that will help us to deploy these resources in areas that are very far away from medical centers. They will be cheap so that we'll be able to do this high throughput screen of different sources. Like, for example, here I would test the water, I would test from the skin of the kids, from the sweat, and to see what's going on. And then, while working especially with the microbiomes, it would be really exciting to follow their microbiome development, especially if we think that the first 3 years are really crucial for establishing the microbiome in kids. So those 3 years overlap with the use of diapers. So it's saying, wow, this is really a great opportunity to use the diapers that already have hydrogel to keep bacteria there and to be able to, in a simply and convenient way, to be able to measure multiple biomarkers of disease. So this will bring reliability, specificity to measure really different, many biomarkers so that we can specifically diagnose different diseases. And for this, I propose using this recombinase system, which we can test different biomarkers and trigger a different threshold of concentration. So the idea would be to biocontain the bacteria inside of the diaper. So this would be safe for the baby because the baby will never be in contact actually with the bacteria, and they will be biocontained. But they would allow an immediate detection and even a peak detection, for example, of even intestinal gases. So we'll be able to monitor the whole microbiome development. So what I'm thinking of would be that during the first 3 years, we'll use this diaper, and then even later, we can even use a multi-diagnostic toilet. And with a lot of basic research in which we need to characterize all the biomarkers that actually determine the development of the microbiome from baby to a child and finally the adult, in which defines this healthy state that might be more than one actually, and to be able to identify these biomarkers that show when actually the patient is falling into disease state, right? With the idea that with probiotics, especially in babies, that they has been demonstrated that they can actually make a mark, can revert the situation or changes in the lifestyle, diet, or directly with therapeutics, depending on the severity of the disease. So the idea is to measure toxic elements that may come, for example, from water, from the environment, and then metabolic byproducts, and finally, microbiota-derived molecules that are so hard to measure now with the current tools that we have. And so basically what I'm proposing is, like, if we think like when we are growing plants, like tomatoes, we need to have these sticks, like they tutor, that helps to keep the plant growing in the right direction. So what we are proposing here is that with these diapers and the toilet, the smart toilet, that we'll be able to monitor the whole microbiome development of kids. And with this, the exciting part is that we expect to be able to ideally eliminate the diseases of tomorrow, right? Especially all these microbiome-related diseases. If we have a healthy microbiome, actually, we are confident that many of the diseases of tomorrow will be diminished. So with that, I think I will wrap up the big concepts here. I hope that I have convinced you that measuring these transient inflammatory biomarkers is important, and that we can use common cell bacteria, that we have the tools now to engineer, that patients can, in a non-invasive way, ingest this bacteria, get inside of the gut, and be able to measure in situ the chemical environment of the gut. But we can also use other materials like hydrogels that will help to encapsulate them and to localize in particular regions. And this will help to also study more in detail particular biomarkers of disease there. And of course, this will also be complemented with other technologies. We're having, for example, now even other technologies like microclippers that instead of actually using a magnet, we can attach to a particular region. And with this hydrogel, we'll keep the bacteria there for a longer time. And then potentially even release therapeutics there. And so I think with all these tools coming back to the harvest season, I think this is a very exciting time for research in the microbiome, but also in microbial biosensors to use them for the benefit of the human health. And I wanted to finish with this message that the gut is integral to our health. So it's important to keep an eye on the information it provides, and this is starting to get like this black box that is getting to get open, right? So that the information that the gut is providing should not longer be ignored. And with that, I want to thank you. This is one of the pictures I like taking from the bridge that connects Harvard. So hopefully people that will be around here for the conference will be able to see some of it.
Wow, wonderful.
Yeah.
So I love the picture, the photo.
Yeah. So if you're around here, it will be a very a nice place for you to go. Hopefully,
actually, I was there, I mean, this summer.
Yes. Well, this summer. Oh, you know, this was more in the fall now that we have the change of the leaves. So it's not yet like that. So hopefully, probably for the November conference, it will get there.
Sure. Okay, so thank you so much. I mean, oh wow, I mean, fantastic. You know, I especially enjoyed this talk, of course, because I personally have worked on similar project for many years, partly motivated by my chronic gut problems. Of course, I mean, her work is really important because endoscopy is really invasive. And I know it because I had multiple endoscopy myself, the procedures, and they in fact damaged my organ, I mean, my gut organ actually. So I also visited this summer, I mean, as I said earlier, I mean, and I gave a talk at SynLogic, a leading probiotic and gut microbiome company, co-founded by Tim Lu, her advisor, of course. And Eugenia, probably you may be another future founder of a startup company regarding the probiotic, the sensor and the diagnostic and the treatment kind of approach. I also love, I mean, the necessary lead to doing the impossible, right? I need to give a talk, I mean, 1 hour from now regarding the craziest idea I have ever imagined. Also, fantastic volunteer work for future scientists. Oh my God, wow, wow, wow. I mean, so I guess, I mean, again, I mean, if you there out there, the recruiter or the search committee, catch her, I mean, if you can. So, okay, so any question? Oh, so I guess, I mean, the Stephanie, you could just unmute and then just, ask the question, or I could read your question for, audience.
Sorry, I was trying to find my unmute button. Yeah, no, very interesting presentation, Eugenia. Thank you. I was curious for the ideas that you showed at the end, thinking about the diagnostic diapers and toilets. Do you envision that you would need a panel of different engineered strains that would each detect, like a subset of biomarkers of interest? And, would this essentially require an engineered consortia? Is that the kind of concept that you're thinking about there?
Right. What actually we're seeing is that it's not only one biomarker, and actually the diseases that are now more challenging are not like detected by only one. So we need as many as possible.
Right.
And that's why actually the challenge of how to miniaturize all that. But with the diaper, we don't need that. Like, I mean, we have a whole surface, right? And so the challenge now is actually to see what are the real relevant ones, because still the microbiome development is really an area of research so far. So that's part of what the basic research that I'm planning to do as well.
Great, thank you.
Fantastic. So that's very interesting question. I mean, so I have another question from Ahmed. So thank you for your amazing talk. You mentioned that the engineered cell face a harsh environment in the gut, and it needs, of course, to be adapted to cope. Each host gut environment is different. So how can you make the sensor function and output robust to this?
So yeah, that's a really very good question. So ideally, the way we're thinking of that is that even when each individual is different, we could have groups of people. So for some particular diseases, we need actually to, for example, isolate microbes and see which ones are actually optimized for those chemical conditions. For example, people with celiac disease, the environment is completely different. Also with IBD, and so like probably we could group those people. But then even among those groups, we could personalize having multiple biosensors. So for example, I was sharing this disease stage detector because that's exciting that we can measure different thresholds. So it happens to us right now that we test, for example, in mice, and the one that works is sensor 2. But when we test in pigs, we need sensor 1, because the levels are different. So we expect, so for research, it's actually useful because like you don't want the whole project to collapse because you didn't get the right concentration, right? But for patients, we are envisioning the same situation, that we will have like an array of different thresholds of the same biosensor. And if sensor 1, 2, and 3 doesn't work, probably 5 and 6 would work, right? What matters is that you can see the change over time. So that same patient, if there's inflammation is getting triggered, you will see an increment later.
Okay, fantastic. I guess, I mean, I have another great question, I mean, from anonymous the attendees. Thank you for your talk. What are your thoughts regarding the challenge of engineered bacteria evolving within the environment that you are using them? Do you have strategies to prevent this? Top question, I guess.
Oh yeah, so if they are already inside, it will keep evolving. Yes, so for this, particularly for the ones with the wireless device, it's something that actually the time is limited. Like it happens like, so it will move with the trajectory that will go along the gut. So we don't think that would be enough time to evolve and to mess the whole thing. I think that is a challenge for people who are working on graphing the bacteria and the idea to keep that for many days, right? So in our case, because they are contained, we don't think that would make a big difference. For those that engraph and would be there for longer time, I think it is a concern, and that needs to be just to see ways that we can actually get rid of that. So I think, for example, with the magnet, though for the magnet for patients has some challenges, so that's why I'm also thinking more of a gripper, for example, but it still should be something that you can actually release, if not, right? The general problem would happen actually the opposite, that they will stop working. That's kind of the main concern. Yep.
Sure, that's excellent. So another question from Justin. Do you see the future project combining your sensor with an in situ modulator of biomarkers? That's interesting. Actually, I want to hear your thought. What would be the challenges in combining the two?
So, modulator of biomarkers. So I'm not sure. It's more about like in the therapeutic side, the idea that the biomarker would release a particular therapeutic.
My guess is, I mean, the Justin asking, sensing and then actuating, combining. How
right.
you're gonna combine, and then what's the challenge, combining that too.
Right.
So you kind of mentioned, and that's also my dream. I mean, I want to do that.
Yeah, and I think, well, the Lu Lab is very actually focused on that. So yes, that is what we are also excited about it. The issues there. So I think the diagnosis is the first step. So first you want to diagnose, to detect, and then to then have the actuator of releasing the therapeutic. The challenge there is that in that environment, bacteria may not be able to produce the high quantities of, for example, a therapeutic protein unit. That's actually the real challenge. But even I think it is doable, and we have already some projects that have been showing that. But for me now, the challenge is that you can do it for some target things, right? But the problem is that we are too late. Because once actually we get already the disease, is too late. We need to be able to see, not even when the bacteria in the stool changes, we need to see it even before. That's why we're proposing this thing of being able to measure small molecules to see the chemical environment, and to be able to detect it, for example, after antibiotic treatment, to see if the chemical environment is changing, so that we are not then like years, when everything is changed, and it's very hard to reestablish the original healthy situation.
Yeah, I guess, I mean, that's everybody's dream. I mean I'm talking about the probiotic engineering or so called microbiome engineers. So any other question? Anyone? Any comment? And actually, I have one question. I will ask more questions, scientific question for you, I mean, later on. But may I join your journal club?
Yes, actually. Yeah. We are now looking for volunteers to speak. And actually, so the volunteers are now named as fellows, so journal club fellows. And so they are also, we have a webpage, so that is also to give visibility to young researchers. So I'm getting inspired by you.
Sure. Okay. So you can please send me email about the detail. Yes. I'll happy to join and then to participate as the. I need to read papers nowadays. I so busy, so I cannot read the paper. If somebody forced me to read the papers, so I wanted to read some paper, and rather than just skimming the abstract and the title. I mean, that's what I'm doing. So unfortunately,
actually, that was Eric Allen was with that issue. It's like, I really want to be. I mean, we don't have time. So like, this was the opportunity, because like we show the papers that are interesting, and also from people from different fields. So what gets excited for someone like a medical doctor, and then like we kind of move around, and it's nice to really be knowing most of the latest papers, right?
Okay, wonderful. So please send me email about those things. And I don't believe I see any more question. Oh, let me see. I have one. Oh, I have another one.
Yeah.
So Ahmed again to ask question. This is amazing technology indeed. Is there any thought to engineer multiple sensor for specific diagnosis? And if so, will there be a problem? Multiple synthetic circuit in a single cell could affect each other's function. Okay, tough question again.
Yeah. Yeah, actually, so I thought that actually these would be helpful. So this one actually for me is what summarizes the big picture of it, because we really want to have this technology for diagnosing many diseases, right? And so the idea here is that first you develop the different sensors, and it's true that sometimes one particular circuit still, synthetic biology, we are still developing our field, so we still are trying to see how to optimize the resources of the cell. So the possibility of having multi-array is, the idea is like not to overload the needs for one particular cell. So like doesn't need each cell to measure everything, right? So the idea is that each cell, each bacteria can measure different things, but then you will be able to test these different circuits in vitro and then in for example, mice models, right? Which, if not with the embed technology that ARL has developed, is something that, you don't want to have that only array testing in pigs. You want to go through the other models as well. So the recombinant system, what allows is to use it in small animals. So I think that's the exciting part of it. And then, yes, this is also like a circle, right? Like once we get these biomarkers, then you can use like logic gates and say, for example, oh, if you have high nitric oxide, okay, what else is triggered, right? So you can use an AND gate and you can catch other molecules that probably now no one have noticed, right? But then you can say, okay, this is also a biomarker. So it will be biomarker discovery. This platform will also allow for that.
Okay, great. So now I don't see any more question. Let me check one more time. So no open. Okay. So I guess we are already past the 11 my time. So I'm going to conclude. So thanks all for joining and staying today again. Next week we'll have Dr. James Liao, the president of Academia Sinica, Taiwan, and member of NAS, NAE, NAI, of course, all three, and another James from Tufts University. See you next week, the same day and time. The follow-up Zoom chat actually will occur via the link in the chat box here. So just click that one and then I will meet you there in a minute. So see you soon, and thank you all for joining again. Thank you. Okay, oh great. I really enjoy. So if you click the chat, the link over there,
Yes.
and then you will basically move to the another chat, and then I will just meet you there in a minute.
Okay, that's nice.
Okay.
Yeah.
So you found the chat link?
Everything's good. Let me see. Yep.
You found it?
Yeah, I click it. Let me see. Oh,
Okay.
here. Yep, I'm here.
So
Awesome.
click, and then once you kind of say change the Zoom, and then you will be in the another.
Awesome. Yep.
Okay. See you soon.
See you. Bye.
Bye bye. Thank you, Tony.
Tony stepped out. I took over for a minute, so.
Okay, so please, post the recording, the YouTube recording, and then I need to post that on later on.
All righty.
Okay, thank you so much.
Have a good one.
You too.